Hello everyone. Today I will be discussing with you all the physiology and biochemistry of molting. In the physiology part of today's topic, I will be discussing the changes and the processes happening in the integument associated with the process of molting. While in the biochemistry part, I will be dealing with the chemical structure and the reactions of sclerotization, tanning, and the hormonal control of molting. Now, let us firstly rebrush our basics and see what molting actually is. So, molting, it is very prevalent in the animal kingdom, and the insects, they are one of them. Molting is responsible for the growth and maturation of a nymph or a larva to finally and gradually an adult. We all know that it is a hormonally controlled process involving the prothoracicotropic hormone, which stimulates ecdysone, which is the initiator of the molting process. We will study in detail in the upcoming slides that how molting, which is the shedding of old cuticle and formation of the new cuticle, how these processes actually take place. Before going on to the main molting process, let us visualize some of the changes in the epidermis which happen before molting. These are namely changes in the cell density and changes in the cell shape. Firstly, look at this diagram of the insect integument. Now observe the epidermal cells where I have pointed. Now look at this figure where the epidermal cells have begun dividing mitotically and exuvial or ecdysal space has started to form due to pressure exerted by the increase in the number of cells upon the existing cuticle. Also, the hemimetabolous and the holometabolous insects, they show some differences with respect to change in cell density. In case of well sclerotized hemimetabolous insects, as we just discussed, firstly the cell density increases due to mitosis before molting. Then the cell density decreases upon cuticular expansion. And after that, cell density returns to the original value as total area of epidermis it increases and the new epidermal cells spread out. This is the reason why the newly formed cuticle is always larger in surface area than the previous one and is the basis for growth in an insect. As we all know, since the larval forms, they have a softer and more flexible cuticle. So when the cuticle unfolds, cell density decreases during feeding because cells they spread out. And afterwards, it increases due to mitosis. Then when the cell growth occurs before ecdysis, as the cells take up more space due to enlargement, cell density decreases and the number of cells per unit area decrease. At the larval to pupal molt at the last, the larval phase shortens due to rearrangement of the cells, which causes increase in the cell density. After density, we have changes in the shape of cells. For instance, in assassin bugs, which are holometabolous, the epidermis is columnar immediately after molting, but becomes squamous until the insect feeds. However, in the holometabolous forms, it is the opposite. The epidermal cells, they are squamous immediately after molting and turn columnar at the time of mitosis, especially before pupation when the larva shortens. Another example for change in the cell shape is of Calpodis, which in its last instar larva develops basal processes. As you can see, these are the basal processes which are developing at the end of cells. And these basal processes have become longer as the diasteroid titer rises. And then these basal processes start shortening as the pupation nears. And also you can see the cells, these are first columnar in shape, they are squamous in shape, and then they elongate to form this columnar type of cells afterwards. Now let us review the events which take place during molting. These are the hormonal stimulation, then apolysis, then secretion of molting fluid, formation of outer epicuticle, formation of new inner epicuticle, deposition of new procuticle, secretion of wax layer, secretion of cement layer, ecdysis, expansion of new cuticle, hardening of the cuticle. The molting process begins when the epidermal cells are exposed to 20-hydroxyecdazone, the insect molting hormone. The hormone enters the inner epidermal cells, which stimulate genes related to molting and formation of a new cuticle. The activated epidermal cells, as you can see here, undergo mitosis, and thus a new larger cuticle is formed later on. The existing structural cuticle, it separates from the epidermal cells, which is known as apolysis, and results in the formation of an ecdysial space. The ecdysial space is then filled with molting gel or granules, which contain inactive chitinase and proteases. And this molting fluid, can be basically of two types: the gel, which is found in the Lepidopteran insects, and granules in case of Columbola order insects. The new inner epicuticle and the outer epicutical layer of cuticle are secreted. And this cuticulin layer protects the newly forming cuticle and the epidermis from the digestion of enzymes by the molting gel. Now the molting gel or granules are activated and converted into a fluid, and the enzymes begin to digest the old endocuticle. And as much as 90% of the chitin and protein breakdown products from the old endocuticle are reused to form a new procuticle like this. The digestion of endocuticle continues until it reaches the old exocuticle, and the old exocuticle, it is resistant to enzymatic action since it is sclerotized, and the remaining molting fluid is reabsorbed. The wax layer and the polyphenol layers are then secreted by the epidermal cells. Just before molting, cement layer is released like this by the dermal glands. And molting, which is properly called as ecdysis, occurs when the exuvium consisting of the old epicuticle and the exocuticle is shed off. After escaping from the old cuticle expands before hardening, either by swallowing extra air or water. Then bursicon is released, which stimulates the procuticle to undergo tanning and sclerotization by polyphenols and be converted to the new exocuticle. During the time between the molts, new endocuticle is deposited continuously, and the cycle, it starts over at the next molt. Now let us understand the different pathways of cuticle hardening. First is the quinone tanning. It is the process responsible for the conversion of tyrosine to quinone, which further combines proteins to form dark sclerotin. This process is controlled by bursicon and 20-hydroxyecdazone, but in vitro tanning of these hormones hasn't been yet possible. These are the reactions which occur in the quinone tanning and subsequent sclerotization. On the top left corner, the structure you see is of tyrosine. The amino acid tyrosine is oxidized by phenyl oxidase, which leads to positioning of this OH group on the carbon atom, and dihydroxyphenyl alanine DOPA is formed later on. Then enzyme DOPA decarboxylase removes this carboxyl group at this end, and then an intermediary is formed which reacts with acetyl CoA in the presence of dopamine acetyl transferase, causing transfer of acetyl group from acetyl CoA, as you can see here, causing the formation of N-acetyl dopamine, which is the fourth product you will see. Then the diphenol oxidase oxidizes the molecule and quinone is formed, which is the fifth product, and the product on the bottom right corner of this slide. And when upon subsequent additions of proteins, finally dark sclerotin is formed at the end. As you can see that NH2 protein is being added here 2 times, which leads to the darkening of the sclerotin. But when the N-acetyl dopamine, it doesn't oxidize to quinone, colorless sclerotin is produced subsequently, and this pathway is known as the beta sclerotization. So let us summarize the pathways I just mentioned. First one was in which the N-acetyl dopamine, which is the pathway in the downwards direction. And in this, the N-acetyl dopamine is being converted into quinone and subsequently into sclerotin upon addition of protein. In the other pathways, the N-acetyl dopamine under the action of desaturase in the topmost reaction removes 2 hydrogen molecules, and as you can see, an unsaturated bond is formed here. This product is thus known as dehydroN-acetyl dopamine. Now this oxidizes under diphenol oxidase, and on addition of protein forms colorless sclerotin, or can also follow other combinations of pathways like this, where it combines with the N-acetyl dopamine. Now let us see the hormones involved in molting, and their structures are as follows. First we have ecdazone, which is the initiator of molting, and it is a steroid hormone. Second is the eglosion hormone, which initiates ecdiesis and is a peptide hormone composed of these amino acids. Then is the juvenile hormone, which causes the retention of juvenile characters. Then we have bursicon, which causes sclerotization. Lastly, we have corpus allatum and corpus caardiacum complex, which cause wax secretion. Now let us see the production and flow of brain hormone. The neurosecretory cells, they produce the brain hormone which flows into the corpora cardiaca and then the hemolymph and then enters the prothoracic glands and corpora allata, where it comes to be known as the prothoracicotropic hormone. Now the PTTH, it stimulates the prothoracic glands to produce ecdysone, and the corpora allata, which produces juvenile hormone which controls the prothoracicotropic gland and the type of cuticle to be formed. That is if the juvenile hormone it is more at the time of molting, larval cuticle is formed, and if it is less, then adult cuticle is formed. Lastly, we have insect growth regulators being used as insecticides, which are in a way a practical application of our understanding of biochemistry and physiology of molting. First, we have polyoxins, which are structural analogues of UDP and acetyl glucosamine and interfere with chitin deposition by inhibiting chitin synthetase. Then we have diflubenzuron, which is a substituted urea and inhibits serine protease, which would activate chitin synthetase and thus preventing the formation of polymerization of chitin. These are the sources that I have referred, and I have taken the animations from this video, molting of the insect cuticle. I will be mentioning the link in the description. Thank you very much. The epidermal cells